Okay. I am going to present some of the results from my PhD project on spatial analysis of some hemiparasitic plants and a predominant host. And if you're on Twitter, you can follow me on that name. I'm a dual student enrolled at 2 different universities. So this is the outline for my presentation, where I will come with the results to answer some of the aims I've set on mortality of a predominant host, Acacia ligulata, and also study the above-ground biomass. But first I will give a quick introduction into parasitic plants in Australia and methods for spatially analyzing them. So there are a great number of parasitic plants globally, but we also have a fair share here in Australia. I've listed a little more than half of the species in these tables and highlighted those species in bold that have been studied together with hosts. So not really that much research that has gone into it. On the left, I've listed some root hemiparasites, and you might be familiar with the top one. That's Australian sandalwood, which here in South Australia is considered vulnerable, if not going towards endangered. And I've been studying that, and also the Acticarbus leafless ballad. In the middle, we have a list of 2 lists of mistletoes. There are plenty of those here in Australia. And on the right, I've listed some parasitic vines that are also widespread, and I've been studying the top one, the large dotted laurel. So one way to analyze the relationship between species is by using spatial analysis, and generally you can divide the data into 3 categories. Quantitative data, where each point within the area has a quantitative value. Then categorical data, where the points are divided into categories within the area. And then spatial data, where you study points and the relation between the points within an area. That could be like plants within a field site, and I've been mostly using the latter one. I'll mention a few methods. So summary statistics is a way to quantify the statistical properties of a point pattern, and there are methods both for local investigations and global investigations. Some global ones being the G function and the F function, or the nearest neighbor, where's the nearest neighbor, and the empty space, how is the empty space between the points, and you can combine those 2 into the J function. Globally we have the Ripley K function and the pair correlation function, the first one being cumulative over the range and the other one being non-cumulative. So you have different options to investigate point pattern. We also have random labeling, which is a good way to create a non-model where we contain the point pattern, so where the points are within the area, but where we shuffle around, let's say, where live and dead trees are within the site. And I'll be using those. A good way to model point patterns by spatial point processes, but I won't come into that much, but it's a good way to analyze the spatial point pattern data. So the aims of my project, or some of the aims, was to see how the mortality was of the Acacia ligulata, both interspecifically, but also in combination with the root hemiparasites and the shoot hemiparasites, and then how the above-ground biomass of the predominant host is spread around within the area in relation to the root hemiparasites. I listed or shown the species I've studied of the parasites here on the left, and this is a picture of the acacia or the June wattle, and this is a photo of an illustration of my field site, and the points within the field site are the June wattles within the field site. And in addition to the locations, I also measured the height and the stem circumference, and I estimated the canopy volume of all the June wattles within the site. And this is a image or a figure of all the trees recorded. So you have the Dune wattles, both live and dead, and then where all the root hemiparasites are, the red ones, and where the parasitic vine was found on the Dune wattles, that the light green. So a lot of points, and this enabled me to do the point pattern analysis of this study site. I first looked into Dune wattle and just looked at that pattern, and as a proxy for age, I used the stem circumference, and this shows 5 generations, and you can kind of see from on the contour lines that after a certain number of generations it seems to be somewhat conserved, or the pattern kind of crystallizes, and it might be due to facilitation between intraspecific facilitation. It could also be where the soil resources are, but I didn't look into that. But I did see if the elevation could be considered covariate for the point pattern, but I didn't find that to be the case. And then looked at the mortality for the Dune wattle using the live and dead pattern, and using the J and K functions. And the gray bars are simulations envelopes to see how it looks on the randomness. If there were no dependence between them, the black line would be in this area. So there seems to be seems like the dead trees, the location of the dead trees seem to depend on the locations of the live trees, or shrubs. And then I went on to look at mortality in relation to the root hemiparasites using a method called tree variate random labeling. And again, we have like the simulation envelopes on the randomness, and at specific distances there seemed to be a higher mortality than would be expected on the randomness in relation to the root hemiparasites. I also looked at the mortality for the parasitic vine. I did it by using a tessellation method for the area of where the Dune wattles were located, and then I used a Monte Carlo procedure to find the number of dead shrubs that would be expected on the randomness in the tiles where the parasitic vine was located, and I found that to be 88, which was somewhat lower than was observed in the window. And further analysis gave strong indications that death was related to the presence of the parasitic vine. I also used the tessellation for the above-ground biomass allocation, and you can see the locations of the root hemiparasites indicated by circles, and seems to be just outside the areas of the high biomass of the host, but still not in areas where the above-ground biomass is low either. So it seems to be kind of perhaps a Goldilocks case again. Like they want to be in between. And looking further into this, using a distance function of the root hemiparasites, we see the same result. Like this indicates the distance from the root hemiparasites. This is the distance or the density of the host, and this is the density including the biomass. So they seem to prefer not to go too close to the host, probably because, or it could be due to they're not as good as competing for sunlight, which has been shown for other kinds of parasites, not for woody parasites. So coming into the relevance for this, a similar or related species of the parasitic vine has been investigated here at this unit to see if it can be used as a biocontrol agent to fight invasive species, and there are some suggested findings that it can actually be used for such a purpose. And I've shown the geographic distribution of this figure. from this paper, and you can see the distribution of the species are investigated on this figure, which is more spread towards the west. So if this shows to be a good biocontrol agent, then perhaps the species I've been investigating can be used in other areas of South Australia and perhaps in Western Australia as well. So for the sandalwood, as I mentioned, it's considered vulnerable, if not endangered, and we need a mix of both political and environmental solutions to counter that. And perhaps the spatial analysis I've been doing here can be helpful for optimizing revegetation efforts. It needs to be tested in other sites as well, but it could be a way to find what a healthy populations of sandalwood would like in a natural community. And it could be perhaps included for other kinds of Santalum, perhaps northern sandalwood in the northern part of Australia. So I would like to thank my supervisors and other people that have helped out, and 3 of my supervisors are present here today. Very grateful for that. And again, if you want to, you can follow me on Twitter, tweet about my research. Yeah.
Thank you very much. I'm sure that everyone's keen to go to afternoon tea, but we do have time for a few questions. Do you have anything they'd like to ask Bjorn? Yes.
I'm blown away and impressed about how much work must have gone into collecting all of the point data in your...
Yeah, it took some time.
So thinking about that, I'm wondering if the Coonamoor plant records, the arid zone Coonamoor research station, have permanent vegetation plots where the species have been recorded, and now a long history of that, and quite a few of these species I think probably occur there.
Yeah.
I'm wondering if that might be another avenue for investigating some of these things, because it has the time connection as well.
Yeah, that's the thing. This is ecological project. So yeah, 3 years is not that long in this regard. So yeah, that's a good input. I would like to follow up on that. So thank you for. Yeah.
Do you have any other questions? Okie doke. Well, I'd like to thank all of our speakers this afternoon. I'm sure they'll